Right. Hi everybody, welcome back again. In this video I want to begin topic 3, and obviously topic 3 will begin with topic 3.1, and that's called Introduction to Biodiversity. Alright, let's begin straight away just by looking at the key terms in the syllabus. It looks like a lot, but I think there's not that much in this topic because it largely relates to 3 or maybe 4 different definitions more than anything. And you can see those definitions above my head already. So first of all, it says that we need to talk about what biodiversity means. And then secondly, it says we need to split biodiversity up into 3 different definitions. We can talk about biodiversity in terms of, well, as you can see above my head here, number 1, species diversity, which is the distribution and the number of different species that you can sample. We've pretty much looked at that already in topic 2, actually. And we looked at the measurement of that in terms of using Simpsons index. Down here it says we need to talk about habitat diversity, which is another measure of biodiversity. Obviously that's just the distribution of different types of habitats within biomes. And then finally, something that we can't really measure at IB level, maybe university level, is the idea of genetic diversity. The diversity of genes within a gene pool. I'll talk about what that means, but it's not really something that we'll be measuring. Apart from that, we need to look at how we can use indexes to compare biodiversity. The one we really look at here is Simpsons index, which we've looked at already in topic 2, but I will give you a little reminder about how you actually use Simpsons index. And then finally down here, well, maybe we can talk about this more in the classroom in our discussions. It's how we can quantify biodiversity and then use it to help conservation efforts, and how biodiversity is affected by human actions, and how a diversity index can help us to look at the severity of human interactions with the environment, which is usually not a good thing. Under applications and skills, distinguish between biodiversity, species diversity, habitat diversity, and genetic diversity. Hopefully you'll be able to do that after I give you my definitions. Look at relative values in terms of biodiversity data, specifically Simpsons index. We've done that quite a lot already in topic 2. Very relevant to paper 1 in the exams, that kind of information. And then finally, discuss the usefulness of using numerical values for species diversity in order to understand the nature of communities and conservation. Quite a lot of repetition there, but really it just boils down to those 4 definitions I have here. What is biodiversity? What is species diversity, habitat diversity, and genetic diversity? Okay, so let's first of all talk about biodiversity. I can tell you a few things about that. Number 1, it was a term that was coined originally by probably the most famous living biologist at the moment, which is Edward Wilson. He came up with the term in the 1980s and kind of did a lot of his work around that. And biodiversity it's more than just measuring the number of species that you can find in an area, or the number of species per square meter unit area. It's the number of species present, which we refer to as species richness, but also the relative abundance of species in that place, which we refer to as species evenness. This relates more to species diversity more than anything else. But as you can see here, as I've already said to death already, we can measure biodiversity in terms of 3 things: species diversity, which is the one we normally look at, habitat diversity, or genetic diversity. Species diversity, as I've already said, is a measurement of species richness and species evenness. It's possible to have a lot of different species present in your sampling, but one species might be very, very overrepresentative because it's very dominant in that place, in that area of sampling, and therefore you can have a lot of species present, but still not have high biodiversity because of the lack of species evenness. Areas of high species diversity are, sorry about the spelling mistake, are usually places that are undisturbed by human action. Humans, as we already know, are very good at reducing species diversity. At the start of the course, we looked at the 5 ways that humans affect biodiversity or reduce it. Hope I can remember this now off the top of my head. Number 1, habitat destruction, habitat islandization, introduction of alien species, overharvesting, and pollution. Pollution being probably the most insidious because now it occurs on a global scale. If disturbances happen on a very localized scale, then it's always possible for ecosystems to recover. If disturbances happen on a global scale, as is happening now specifically with human-led climate change, then those habitats can't, or those ecosystems as a whole can't recover. I've said here that Simpsons index is our most common measure of biodiversity, and I will give you a reminder of that, as I said already, at the end of this little talk. Number 2, habitat diversity. Very, very straightforward thing to understand. It's just a range of different habitats in an ecosystem, or maybe I should have said in a biome. Of the different habitats that we look at, the different biomes that we look at, the most biodiverse in terms of habitat diversity are probably rainforests. And the least diversity in terms of habitat diversity are desert and Arctic biomes. Very little in the way of different types of habitats, and therefore very little in the way of different types of niches to allow different species to exist. And then number 3, genetic diversity is the range of genetic material or genes present in a population, species, or a specific gene pool. Genetic diversity is promoted by having large number of niches available, but it's also promoted by having isolated habitats. If you have isolation, then you encourage speciation by evolution. So those are the 2 ways I would say that genetic diversity is promoted. Okay, and getting towards the end of this, in terms of measuring biodiversity, well, we can measure species richness, we can measure biodiversity, and we usually do that in terms of Simpsons index because it gives us a measure of both. Remember, please, that Simpsons index does not have a unit. The word index means something without a unit. It's just given by this equation, or at least this is the version of Simpsons index that the IB prefers us to use. D, or Simpsons index, equals the total number of organisms that you find multiplied by itself minus 1, divided by the total number of individuals found of one species you're interested in across all of your quadrat sampling, multiplied by itself minus 1. I won't go into that now, but if you can't remember exactly how to use Simpsons index, please can you go back to topic 2 and just go back over that again. I stole this map from another website, I should say. This is a measurement of global biodiversity, specifically plants, or species diversity measured in different biomes. I've already said that the biomes with the least species diversity are Arctic biomes, the north and the south, but also desert biomes, for example the Sahara here. That's why they have the lightest colours, yellow colours. Highest biodiversity across the equator, more or less, specifically associated with rainforest. Probably coral reefs are the only other ecosystem that can compete with tropical rainforest in terms of overall species diversity. But also you see the red points. The red points are hotspots of species diversity. One example that we've looked at already is fynbos in South Africa, which is, for many different reasons, is a global hotspot of biodiversity, specifically for plant species. Okay, and then finally, just to finish off, why exactly is this these different patterns of biodiversity, or specifically species diversity across the planet? Well, it happens where you have the least limiting factors. Limiting factors are abiotic factors that, as the name suggests, limit growth or productivity or in terms of this biodiversity. If you have fairly high rainfall, a high amount of sunlight availability, and a fairly equitable temperature, those are all factors that promote biodiversity. And that's associated with high primary productivity, range of soil types available, long periods of uninterrupted succession, so you can get to a number of different climax communities, lack of plagioclimax, as we've talked about before. And specifically, if you have these kind of conditions and a lack of limiting factors, you promote many, many different niches to become available. We associate rainforests with high niche availability. I've said here that occurs not only horizontally across land or land masses, but also vertically in terms of altitude, and in rainforest in terms of distance up the canopy. Okay, so that's a very, very brief, but I hope fairly accurate introduction to what biodiversity means. Obviously, we talk about this more in the classroom. Please can you go back over this and make sure you're happy with it before we continue with topic 3.2 and move on to conservation. Okay, thanks everybody, take care, and I will speak to you in the classroom.